Welcome to Hypersensitivity. We will be discussing the 4 types of hypersensitivity and what hypersensitivity overall means. Hypersensitivity reactions can be defined as a heightened state of immune responsiveness that can cause host damage. And we're talking cellular damage, which can lead to tissue damage, which can lead to the organism damage, and eventually death. Typically, this is an exaggerated response to a harmless antigen that can result to injury to the tissue or disease and even lead to death. Our favorite immunologist, Gale and Coombs, devised a classification system for these reactions based on 4 different categories. So you should note on this slide that hypersensitivity is an exaggerated response to an innocuous antigen. That means an antigen that under normal circumstances is harmless. The 4 categories are type 1, which also is anaphylactic, type 2, which is cytotoxic, type 3, which is immune complex, and type 4, which is cell-mediated. Types 1 through 3 are considered immediate, and type 4 is considered delayed. So that's very important to remember. Types 1, 2, and 3 are considered immediate, and type 4 is delayed. Type 1 reactions, these occur when the cell-bound antibody reacts with an antigen to release physiologically active substances. Now, where do we have physiologically active substances that could be released? I'm hoping you're thinking about our granulocytes. Complement is not involved in type 1 reactions. Now, as we go through these reactions, it's important to know which antibody is involved, IgG or IgM, and whether or not complement is involved. It's important to know the type 1, 2, 3, and 4, and also the common name, and also the protective role that each one has. So type 1 is also known as anaphylactic reactions. So these are the ones that if people are allergic to bee stings and they get stung and they quit breathing and they could die, that's an anaphylactic reaction. Type 2 reactions are those in which free antibody reacts with antigen associated with cell surfaces. So this is important to remember. Type 2 and 3 are easily confused. So type 2 is associated with an antigen associated with a cell surfaces. Complement does play a major role in producing tissue damage, and these are also known as the cytotoxic reactions. Type 3 hypersensitivity, the antibody reacts with soluble antigen. Okay, so remember type 2, we have cellular antigens bound to cells. In type 3, our antibody reacts with soluble antigen to form complexes and precipitate out in the tissues. Complement plays a major role in type 3 reactions in producing tissue damage, and these are also known as immunocomplex reactions. So think about what's actually happening. You have an antigen that's soluble and floating around in the serum, and it's complexing to antibodies, and those are precipitating out in the tissues, just like we can see precipitates in our reactions in the lab. Those precipitates are falling in the tissues and settling there, and that is what is going on. That's bad. So these are known as immune complex reactions. Type 4 hypersensitivity differs from the other 3 because in this one, sensitized T cells, not antibody, are responsible for the symptoms that develop. So that's very important to remember. In type 4, we're not talking about antibodies causing symptoms. We're talking about sensitized T cells. Complement is not involved in type 4, and this type 4 is also known as cell-mediated reactions. So types 1 through 3 have been referred to as immediate because symptoms develop within a few minutes to a few hours. And type 4 has been called delayed because you don't really see symptoms until 24 to 48 hours after contact with the antigens. All hypersensitivity reactions have in common the asymptomatic sensitization phase. That might be a test question. Now, while you're studying, as you're taking your notes and doing the fill in the blank, to help you study, you might make yourself your own test. So on a separate piece of paper, write the question, what do all hypersensitivity reactions have in common? The answer is the asymptomatic phase, asymptomatic sensitization phase. But what you can do is make yourself out a test later, like write out the questions, leave the blanks, study, and then later go back and see how many you can write the answers to without looking. That is an excellent way to study. Okay, back to type 1 hypersensitivity. We're going to get down into the details of this. Anaphylactic reactions are mediated by mast cells. That's very important. The distinguishing feature of type 1 hypersensitivity is the very short lag time, usually seconds to minutes, between the time you're exposed to the antigen and the onset of clinical symptoms. The key reactant present in type 1 or the immediate sensitivity reactions is IgE. And IgE is made against allergens which bind to basophils and mast cells, causing degranulation, and the system is flooded with histamine and heparin, causing the anaphylaxis. Antigens that trigger the formation of IgE are called atopic antigens or allergens. So an atopic antigen, an antigen that triggers IgE, which attaches to basophils and mast cells, releasing histamine and heparin. So I've said histamine and heparin several times. I think that's important. Atopy refers to inherited tendency to respond to naturally occurring inhaled and ingested allergens, so things that we breathe in or eat, with continued production of IgE, which attach to cell membranes. So let's go back and study. So refresh. What kind of heavy chains or H chains does IgE have? Do you remember? Epsilon. All right. Patients who exhibit allergic or immediate hypersensitivity reactions produce a very large amount of IgE in response to a very small concentration of antigens. Characteristics of IgE are described in Chapter 4, maybe. But let me tell you about two buddies that were the first researchers to demonstrate the factor in serum that was responsible for type 1 reactions. So Kustner, who was allergic to fish, and Prausnitz, who was not allergic to fish. So Prausnitz so Kustner, who was allergic to fish, got some serum from his buddy Prausnitz and injected his serum with the fish antigen, and that induced an allergic reaction at the site. So that's called passive cutaneous anaphylaxis. So I tell you that story not to remember, so you have to memorize who Kustner and Prausnitz was, but so you know that people did some crazy things back in the day and that it can be inducible. All right, although actual antibody synthesis is regulated by the action of cytokines, which we've already covered, the tendency to respond to specific allergens appears to be linked to inheritance of certain MHC genes. So that lets us believe that allergies are indeed hereditary. All right, we can skip over these. Not too important. All right, type 1 sensitivity. Langerhans and dendritic cells internalize and process allergens from the environment, and they complex to MHC class II molecules in lymphoid tissue, where the synthesis of IgE occurs. Binding of IgE to cell membrane increases the half-life of IgE from 2 or 3 days up to at least 10 days. And once bound, IgE serves as an antigen receptor on mast cells and basophils. Cross-linking of at least 2 antibody molecules by antigens triggers the mediators to be released from these cells. And what are these mediators? Histamine, heparin, and some other factors. Okay, so the cross-linking of surface-bound IgE on basophils and mast cells by the specific allergen, like whether it's bee venom or pollen or peanut allergen, that causes a change in the cell membrane that result in the release of the preformed primary mediators such as histamine, heparin, eosinophil chemotactic factor of anaphylaxis. So chemotactic factor, what do you think that means? It calls other cells to the area. And neutrophil chemotactic factor, as well as proteases. So basophils are about 1% of the white blood cell population, and they have granules which contain histamine that have a very high affinity to IgE, and basophils contain 10 times more histamine than eosinophils. So why is this important? Because histamine is vasoactive and causes constriction of smooth muscle, which is found in several very important places, such as bronchioles in the lungs, blood vessels, and intestines. And this also can increase capillary permeability, altered cardiac contractility, and increased production of mucus in the upper respiratory tract. And histamine can also cause the redness of skin called the wheal and flare, and wheal is spelled W-H-E-A-L and flare, F-L-A-R-E. In addition to immediately release of preformed mediators, these mast cells and basophils are triggered to synthesize other reactants from the breakdown of phospholipids in the cell membrane. Arachidonic acid is released from the breakdown of phospholipids and causes pain and inflammation. And arachidonic acid, it's very similar to what's found in hot peppers. So can you imagine like Tabasco being dumped on your inside your body? That's what it's like. Okay, so type 1 hypersensitivity. These products are responsible for late phase allergic reactions seen within 6 to 8 hours after exposure to the antigen. And newly formed mediators include the platelet activating factor. So we can just stop right there. So we can see that hypersensitivity and anaphylaxis, so that can lead to an irregular heartbeat, decrease in breathing, increased mucus production, our veins and arteries constrict, and then on top of that we get activation of platelet activating factors, which can start a coagulation event in the body. So it's very serious. Anaphylaxis is the most severe type of allergic response because it is an acute reaction that simultaneously involves multiple organs, and it can be fatal if not treated promptly. And the symptoms of the anaphylaxis depend on the route of exposure, like whether it's inhaled or injected or eaten, dosage, so how much did you breathe, inhale, or eat, and the frequency of exposure. So a test question might ask, what does the severity of anaphylaxis depend on? And there's about 4 things. Anaphylaxis severity depends on the number of IgE-coated mast cells or the buildup of IgE from previous exposure. 2, the massive release of histamine. 3, bronchoconstriction, and 4, irreversible shock or swelling. Rhinitis is the most common form of atopy or allergy. Food allergies are another example of type 1 immediate hypersensitivity. And asthma can also result. The airflow obstruction is due to bronchial smooth muscle contraction, mucosal edema, and heavy mucus secretion. So type 1 causes can include drugs like penicillin, bee or insect stings like wasps or bees, hornets, and then foods like peanuts, shellfish, or dairy. Not plants or poison ivy. Those fall in type 4. And so for these exposures and the allergic reactions, what is the immune mediator? The answer is IgE. So write that question down as one of your self-test questions. Type 1 hypersensitivity, it has a protective role against parasites. So I told you the protective roles were important to remember. Testing for allergies or immediate hypersensitivity can be categorized as in vivo, which means in the body, or in vitro, which means in the lab. In vivo methods include the direct skin testing, which is the least expensive and most specific. So in vivo, this is done inside the body. So this is the scratch test or the cutaneous test or the prick test that you've probably seen pictures of or perhaps even experienced. And the in vitro test measure either total IgE or antigen-specific IgE. So in vitro, outside the body, this is less sensitive but better for the patient because it's less traumatic. All right, type 1 hypersensitivity, the first test developed was for the measurement of total IgE, which is a competitive radioimmunosorbent test. And in the wrist, they used radio-labeled IgE to compete with patient IgE for binding sites on a solid phase coated with anti-IgE. So if a test question might ask, what are the two tests used to determine type 1 hypersensitivity? And those are RIST and RAST. So RIST was a competitive IgE, a competitive assay for total IgE. And RAST is for the specific IgE. So RAST was the original commercial testing method to determine specific IgE, RAST. So RAST measures specific antigen. So if you wanted to know if you were allergic to shellfish or peanuts, you would need the RAST. If you just wanted to know if you had an increase of total IgE, you would use the RIST. So there will be a test question that asks if you needed to see if a patient was allergic to a specific allergen, such as penicillin, which test would you choose? I hope you would say RAST. All right, the principles of the test are not so important right here, although there is a diagram. So remember, RIST measures total IgE. It's competitive. It could be competitive or non-competitive. RAST measures antigen-specific IgE, and it is non-competitive. All right, now we're going to move on to the type 2. In type 2, remember that antigens are cellular. This is important because in type 3, they are soluble. Type 2, cellular. So let's remember that they go in alphabetical order for type 2 and type 3. Type 2, C for cellular comes before S for soluble in type 3. The reactants responsible for type 2 hypersensitivity or cytotoxic hypersensitivity are IgG and IgM. The antigens responsible for their formation may be altered self-antigens or heteroantigens. In type 2 hypersensitivity reactions, antibody coats cellular surfaces and promotes phagocytosis by both opsonization, which means that's like our salt and pepper, and activation of the complement cascade. Then, macrophages, neutrophils, and eosinophils have Fc receptors that bind to the Fc region of antibody on target cells, enhancing phagocytosis. Natural killer cells also have Fc receptors, and if these link to cellular antigens, cytotoxicity results. Once activated, complement may coat cells with C3b, and we go into our complement cascade, which facilitates phagocytosis. In type 2, complement may also generate cell lysis if the cascade goes to completion, and we knew that already. Examples of type 2 reactions include transfusion reactions, which also include hemolytic disease of the newborn and autoimmune hemolytic anemia, including warm IgG and cold IgM or autoagglutinins. HDN or hemolytic disease of the newborn is caused by an Rh negative mother that is pregnant with an Rh positive baby. In type 2 hypersensitivities, some type 2 reactions involve the destruction of tissues because of combination with antibody. Examples of this type of type 2 include Goodpasture syndrome, which is produces an antibody that reacts with the basement membrane protein found in the kidney, in the glomerulus, and pulmonary alveolar membrane, so it's going to cause a kidney or respiratory problem. Also Hashimoto's disease, which is a thyroid autoimmune disease. Myasthenia gravis, which causes the face to droop. It's you have a antibody against your acetylcholine. And then type 1 diabetes, and these are discussed later on in the autoimmunity chapter, which is coming up very quickly. We should also mention the direct Coombs test, which can detect in vivo attachment of antibodies to the patient's own red blood cells. The indirect Coombs test can detect in vitro attachment of antibodies in the patient's serum. So indirect, in vitro, direct, in vivo. So the indirect Coombs can detect in vitro attachment of antibodies in the patient's serum to a panel of reference red cells with known antigens on their surfaces. And the protective role for type 2 is against viral infected cells. Now we're moving on to type 3. Type 3 hypersensitivity, you have soluble antigen that binds with antibody and precipitates out in tissues, causing complement binding, especially in the glomerular basement membrane, vascular endothelium, joint linings, and alveolar membranes. So you have IgG and IgM involved. However, in the case of type 3 reactions, the antigen is soluble. So again, the difference between type 2 and type 3 is whether the antigen is on the cells or soluble. When soluble antigen combines with antibody, immune complexes are formed that precipitate out in the serum. Normally, complexes like these are cleared by phagocytic cells, but if the immune system is overwhelmed, these complexes deposit in the tissues. An example of a type 3 reaction is the Arthus reaction, serum sickness, lupus, rheumatoid arthritis, and autoimmune diseases. If these complexes precipitate out in the tissue, they can bind complement, causing damage to that tissue involved. And precipitating complexes occur in mild antigen excess, and these are the ones that are most likely to deposit in the tissues. Sites in which this deposition typically occurs include the glomerular basement membrane, vascular epithelium, vascular endothelium, joint linings, and pulmonary alveolar membranes. Long-term changes include loss of tissue elements that cannot regenerate and accumulation of scar tissue. And once scar tissue has formed, There is no going back, no getting better. The classic example of a localized type 3 reaction is the Arthus reaction. And this inflammatory response is caused by antigen-antibody combination and subsequent formation of immune complexes that deposits in small dermal blood vessels. So here is a depiction of the Arthus phenomenon. In the Arthus reaction, precipitates of immune complexes in small blood vessels under the skin that occur when antigen is injected in an animal with large amounts of circulating antibody. The Arthus reaction is very rare in humans. Serum sickness is a generalized type 3 reaction that is seen in humans, although not as frequently as it used to be. And serum sickness results from passive immunization with animal serum, usually horse or bovine, used to treat infections like diphtheria, tetanus, and gangrene. This is a reaction to proteins in antiserum derived from animals, usually 4 to 10 days after exposure, and the symptoms are fever, skin eruptions, joint pain, and swollen lymph nodes, but usually asymptomatic. Type 3 reactions can be triggered by either autologous or heterologous antigens, and several of the autoimmune diseases fall into this category. Systemic lupus erythematosus and rheumatoid arthritis are two examples of type 3 testing. So you do need to remember which autoimmune diseases fall into which type of hypersensitivity. For lupus testing, these patients have a high titer of anti-Smith antibody, but the important thing to remember is that in rheumatoid arthritis, there's an antibody called rheumatoid factor, and this is such an interesting antibody. The body, like the body, human body, your own body, produces an antibody against the Fc region of your own IgG antibodies. So the rheumatoid factor is this antibody of IgG against the Fc region of your own IgG antibodies. It's just crazy. Complement enhances tissue destruction in both of those diseases, both lupus and rheumatoid arthritis. Complement both rheumatoid arthritis and lupus. Okay, finally, type 4 or delayed hypersensitivity. Type 4 hypersensitivity differs from all the others in that sensitized T cells play the major role in the problems. Antibody and complement are not directly involved. Very important. There is an initial sensitization phase of about 1 to 2 weeks that takes place after the first contact with the antigen, and then upon each subsequent exposure, symptoms typically take several hours to develop and reach a peak at 48 to 72 hours after exposure to the antigen. So this is considered delayed, 48 to 72 hours. An example of this type of disease is graft versus host disease. So if you have a kidney transplant and the kidney has white cells left behind and it attacks the host, that is the type 4 reaction. Okay, these are the T helper 1 cells are activated and release cytokines, all kinds, interleukin 3, interferon gamma, tumor necrosis factors beta and alpha, and these cytokines recruit macrophages and neutrophils to the area, which produce swelling, clotting, and generally enhance an inflammatory response. Cytotoxic T cells are also recruited, and they bind with the antigen-coded target cells to cause tissue destruction. Allergic skin reactions to bacteria, viruses, fungi, and environmental antigens such as poison ivy or nickel typify this type of hypersensitivity reaction. So you need to know which haptens, these are considered haptens, nickel, poison ivy, that can be in type 4 reactions. And these are things like we're talking about cosmetics, latex, detergents, contact dermatitis will cause sensitization of cytotoxic T cells, causing a macrophage response. So those are all important and could be test questions. Further examples of type 4 hypersensitivity include the contact dermatitis I just mentioned, hypersensitivity pneumonitis, and the tuberculin skin test that uses Mycobacterium tuberculosis antigen injected under the skin. So the TB skin test is also called the PPD, the purified protein derivative. And again, here's thrown in for good measure is a picture of a type 4 hypersensitivity reaction, which is poison ivy. And as a reminder, natural rubber or latex antigens can trigger both type 1 and type 4. So just because it's a type 4 reaction doesn't mean that it can't also cause a type 1 anaphylactic reaction. And there we have the hypersensitivity lecture completed.